So today let us speak about the subject plant and animal biotechnology. And that too, we are going to deal with the animal cell cultures, which is very predominant topic, which is quite useful for all the students who are looking for any careers in related to production units, especially using animal cells. So how is this unit divided is that the first part speaks about culture media, composition and preparation. I have already seen what is the balanced salt solution and the simple growth medium. And the second thing I think we have already discussed about the chemical, physical and metabolic functions of different constituents of your culture medium. And what are the roles of carbon dioxide, serum and protein-free defined media and their applications. And in that line today, we will be looking into culture and maintenance of different animal cell lines. So, what exactly primary and established cell lines mean? So, that would be quite interesting, I feel. You see here, these are the tissue culture flasks. So, those who haven't gained an experience in handling a tissue culture flask, this is how it looks like. Of course, we would handle it in the coming laboratory class, but this is how it looks like for the students who are yet to study the basics of these things. Yeah, so this is how a tissue culture flask looks like, and this is a simple setup of a laboratory, which is shared by Invitrogen, which is one of the famous company which has shared this simple descriptive self-explanatory picture in their portfolio. So if you see here, one could very clearly see that this is a laminar hood, and what all things you should have within a laminar flow is something like you have 70% ethanol for sterilization process or decontamination process. You have your cell culture flasks here, a tube rack. You have your reagents and media, and you have a pipetta, which is electronic. Yeah, and this is nothing but a chair, and you have a waste container, and you have wrapped disposable pipettes down here. Yeah. So this is always a half shut. This is a glass. It's made up of glass, and this is always half shut, so that you do not breathe out your air into the chamber. Okay, in order to keep your uh, working chamber free of contaminations. So here, what does cell culture define? So cell culture refers to the removal of cells from an animal or plant. In this case, we are talking about animals, but however, and their subsequent growth in a very favorable artificial environment. So why artificial? As you know, we are removing the cells from its original position, that is in vivo, from any plant or animal, and you are trying to grow them in an artificial environment, isn't it? The cells may be removed from the tissues directly and disaggregated, okay, either by enzymatic or through mechanical dispersion before you cultivate them, or they may be derived from a cell line or cell strain that has already been established. So what is the meaning of it? Either you can prepare your own cells from tissues directly taken from an animal source, or you can buy a cell line which has already been established. And most of the industries go for the second step, that is to buy a cell line from already established institutes. So whenever you're trying to prepare your cells from the animals as a primary source, it's called primary culture, isn't it? So primary culture refers to the stage of the culture after the cells are isolated from the tissues and are proliferated under appropriate conditions until they occupy all of the available substrate. So here in vitro, substrate can be a plastic bottle, which we have seen in the previous slide. I mean this way. So this can be your substrate. So at this stage, what happens is when the cells have taken their positions within this flask and they have grown all over, okay, so that means they have completely utilized the available space in this flask. Then it is said to be confluent. That means the cells have reached its confluence. So that is the meaning of it. At this stage, once the cells have already occupied all the available space, that means they need to be subcultured. Okay, that means passaged. Okay, this is called passaging of your cells. How do you passage them? You passage them by transferring them into a new vessel with fresh growth medium to provide more room for continued growth. Okay, so what happens when you do not passage? That means there is a lot of space constraints for the cells to grow within this flask. So because of this space constraint, what happens is there will be a lot of contact inhibition. So when there is a lot of contact inhibition, there might be some cells which will trigger the apoptosis because of lack of space. So this is chain reactions. So once a few couple of cells have already started its apoptosis, that means what, programmed cell death, and this signal is being transferred to almost all the cells in your culture. Okay, so once there is no enough space and no enough medium for them to grow, they automatically go into apoptosis or programmed cell death. So if you do not want that to happen, and if you want the cells to be continuously cultured, that means you have to passage them. Okay, so after this first subculture, the primary culture becomes known as a cell line or a subclone. And the cell lines derived from primary cultures will have very limited lifespan. That means they are finite. And as they are passaging, cells with the highest growth capacity will dominate, okay, resulting in some changes in genotypic or phenotypic uniformity within the populations. So what happens when they undergo certain genotypic and phenotypic changes in their characteristics, and they can become continuously growing? That means they can reach immortality, or in other words, they can be defined as infinite. Okay, so against what we are discussing here. So when they have a limited lifespan, they are termed to be finite. And when they do not have a limited lifespan, which is continuous in its growing conditions, that is called infinite. So for you to have an established cell line, your cells need to be infinite. So where does this infinite cell lines have their advantages is when you start a company where you use these cells for producing certain type of recombinant proteins or any other secondary metabolites for that purpose. So then you need cells which are infinite. Okay? So this is what is a cell line. So how do you go on as far as a laboratory is concerned? So this is a very simplified picture which I have displayed here. See here, the cell culture refers to the removal of cells from an animal or plant, as I already told you, and their subsequent growth in a favorable artificial environment. So your tissues can be from mouse, from mammals, can be from embryos, can be from embryonated eggs. Okay, so here you have an organ removed from all these species, and a part of this organ is called an explant, isn't it? So we have already seen what explants are, and these explants are finely cut tissues. Okay, finely it is chopped, and then it is subjected to enzymatic digestion. So in this case, what kind of enzyme we are talking about is trypsin. Why? Because when we are talking about an animal origin, sources or an explant from an animal origin, they have very strong connections because they have a lot of connective tissues which connects every part of your tissues. Okay, so if you need your cells to be independent or individual, all these connective tissues need to be ruptured, and that is being carried out by this enzymatic digestion. In this case, either we use trypsin or collagenase. There are two types of enzymes. Okay, so once your cells have been digested enzymatically, they are grown in a medium. So now there are two types of culturing. One is monolayer culture, the other one is suspension cultures. Okay, so the monolayer culture is nothing but you grow them like this on a flat bottomed flask. Okay, and as the name indicates, suspension cultures means you grow cells under suspension. That means they would be freely floating within a particular medium, growth media. In both the cases, you use well-defined growth media for your growth of the cells. So simply, I have just put down as a flow chart here so you can understand it. So isolated tissue, you have got your isolated tissue, and then you disaggregate them, and then you get your primary cell culture here, which has got limited lifespan. After certain proliferations, they undergo senescence. Senescence is also death, cell death. okay, which is a normal process. Okay, so you have finite cell cultures and you have continuous cell lines if your cells become immortalized, which has acquired the ability to proliferate indefinitely because of some transformations, either genetically or physically. Normally, genetically, they undergo certain type of transformation, which give them the uh, possibility to continuously grow in culture. That is called your continuous cell line or immortalized cell line. So, with this, I think I'll stop this session. So, we will have further discussions on how to maintain the cultures which have now produced either as primary or established cell lines.